In this topic, we're going to discuss the immobilisation of enzymes. So by the end of this topic, you should be able to answer the questions: what does immobilising enzymes mean? What are the uses of enzymes? What are some examples of how to immobilise enzymes? What are the advantages and disadvantages? We're also going to look at the technique of immobilising amylase into alginate beads. And then finally, what's a commercial example of immobilised enzymes? Now, we've discussed how fermenters can be used to manufacture enzymes. These enzymes are used in a wide range of industrial processes, for example, in foods, agrochemicals, and drugs. Now, in many cases, the enzyme and substrate are mixed together to form the product. The product is then extracted, and the rest of the mixture, including the enzyme, is discarded. Enzymes are costly to produce, so this is both wasteful and expensive. Now, because enzymes are not used up in reactions, keeping them for future use is cost-effective. So one way to do this is to immobilise the enzymes so that they can be retained rather than discarded. So here you can see the enzymes in green that have been attached to an inert material. Notice how the reaction of the blue substrate can still occur in the active site of the enzymes. And at the end of the reaction, the product can be collected and the enzyme is retained so it can be reused. So what are some examples of uses of enzymes? Well, tanning uses proteases to soften and remove excess meat. Washing powders use enzymes. For example, lipase is used to break down grease. Protease is used to break down blood on clothes. Cheese manufacture, so using rennin. Diagnosis, for example, dipsticks and biosensors. We're going to discuss these in another topic. Treatment, for example, the removal of embolisms. Now, an embolism is a blood clot that blocks an artery. Immobilisation involves attaching enzymes to an insoluble material so that the enzyme is kept in place during the reaction. So examples include immobilised enzymes on inert glass or in alginate beads. Enzymes can be linked to other materials. They can be trapped in a mesh of an inert material, or they can be held in a semi-permeable membrane. So here you can see the different examples, and the little pink dots represent the enzymes. Can you think of some disadvantages and advantages of using immobilised enzymes or cells? Well, advantages would be that you can reuse the enzymes. They're good for continuous operation. The product is free of the enzyme, and the enzyme's heat and pH stability is improved. You can also develop multi-enzyme reaction systems, and it does reduce the problem of effluent disposal. Disadvantages would be that immobilisation may alter the enzyme shape and function. It is more expensive, and the enzymes may detach. Right, let's have a look at immobilising amylase enzymes in alginate beads. So amylase is mixed with the solution of sodium alginate. The mixture is then dripped through a syringe onto a solution of calcium chloride. The sodium is displaced by calcium ions, resulting in the formation of a hard, insoluble bead of calcium alginate, with the amylase molecules trapped inside. The alginate is left to harden and then it's rinsed off to remove any excess amylase that wasn't trapped. The beads are then used to form a column in a suitable container. And then the starch is trickled down the column and converted to maltose, which is collected at the base. It can be passed through again to remove any starch not yet acted on. The final product, maltose, is enzyme-free, so it doesn't need purification. This technique can also be used to trap microorganisms rather than enzymes. So the process is the same, except you use microorganisms rather than enzymes in the initial mixture with sodium alginate. So an example of commercial use of immobilized enzymes is the manufacture of lactose-free milk for people with lactose intolerance. In this case, the mobilized enzyme is beta-galactosidase. Milk contains the sugar lactose, which is the cause of the discomfort experienced by lactose-intolerant individuals. So to produce lactose-free milk, the milk is passed over the immobilized beta-galactosidase, which catalyzes the hydrolysis of lactose to glucose and galactose. Okay, summary questions. What we've looked at in this topic: what does immobilizing enzymes mean? Can you think of some uses of enzymes? What are some examples of how to immobilize enzymes? Can you think of the advantages and disadvantages of using immobilized enzymes? And can you describe the technique to immobilize amylase into alginate beads? And then finally, can you think of a commercial example of immobilized enzymes? And that concludes our lesson. The end.